Hello, and welcome to the Clinical Care Options Oncology Podcast. I'm your host, Tim Quill. Today's episode features Dr. George Demetri of the Dana-Farber Cancer Institute and Harvard Medical School in Boston, Massachusetts, and Dr. Alexander Drilon of the Memorial Sloan Kettering Cancer Center in New York City. They will be discussing key efficacy and safety data, along with current approved indications for first-generation TRK inhibitors in patients with solid tumors. This episode is part of a larger educational program titled The Expanding Clinical Application of TRK Inhibitors in Cancer Care, a podcast series. For more information on Dr. Demetri and Dr. Drilon, along with a link to the complete program, including a downloadable slide set covering the content of this podcast, please visit the show notes for this episode. Now let's get started and hear what the experts have to say.
Hi, I'm Dr. George Demetri at Dana-Farber, and I'm joined by my colleague, Dr. Drilon. Dr. Drilon?
Hi, Alex Drilon from Memorial Sloan Kettering Cancer Center.
And we're here to talk about some of the clinical data and current indications of TRK inhibitors. If you caught episode 1 where we were talking about finding these patients, now we're going to talk about why we care about finding these patients so much. So Alex, tell us, what are the key clinical trials and the data that led to the FDA approvals of the two available first-generation drugs, larotrectinib and entrectinib?
Yeah, we'll keep the beginning simple and say that these are tyrosine kinase inhibitors, similar to other TKIs that we've used in other oncogenic contexts. And the two major TKIs are larotrectinib and entrectinib. And they differ in that larotrectinib is a selective TKI for TRK, meaning that it inhibits TRK A, B, and C. And entrectinib is a multi-kinase inhibitor. So it inhibits TRK A, B, and C. However, it also has activity against other kinases such as ROS1. And in fact, it actually has an indication for the treatment of TKI-naive ROS1 fusion-positive lung cancers.
You mentioned something interesting there, that these are tumor agnostic except for the ROS1 lung cancer approach with entrectinib. Do you want to comment on that?
That's right. So the approval of these TRK TKIs was actually monumental. It was a seminal approval of the first targeted therapy or therapeutic for an oncogene positive context regardless of cancer type. So the FDA approved both TKIs for NTRK fusion-positive cancers. It didn't really matter what these cancers look like under the microscope. And that goes back to the first episode where we mentioned that these NTRK fusions can be found across a wide variety of pediatric and adult cancers. And that had a bearing on how we designed the clinical trials that led to the FDA approvals and, of course, the approvals in other healthcare environments subsequently of these two TKIs. The design at its core is what we call a basket trial, where the primary enrolling factor is the presence of this molecular alteration, in this case, an NTRK fusion, regardless of what the cancer looks like under the microscope. So tumor agnostic, it didn't matter what the cancer types were. And actually, it was also age agnostic because we had both pediatric patients and adult patients that were treated in both registrational programs.
And that was a great way to develop the drug where the key driver was important. It didn't matter if it was a breast cancer, a colon cancer, a brain tumor, or lung cancer, or a sarcoma. So it was a breakthrough. But for most doctors who think about disease anatomically, I think it still doesn't resonate. I think a lot of people say, oh, I have an interest in breast cancer, or I have an interest in colon cancer. It's like, what do you do with a drug like this that really is a little bit of a fits-all? I think that's interesting.
It is exciting that we're breaking down, if you will, histologic barriers. And other examples include the immune checkpoint inhibitor approvals for TMB high cancers or MSI high that actually preceded the tumor agnostic approval of these TKIs. But I do think that more and more we're going to need to broaden our thinking and be inclusive of such indications, because I feel like at the end of the day, it benefits patients. There's no way for some of these small subsets that we can run a single trial to prove that targeted therapy works. But leveraging this basket trial strategy, we were actually able to be much more inclusive than we were in the past, where we were sort of locked into cancer-specific trials.
And Alex, I think you raised the point about these trials were absolute breakthroughs just because the dramatic nature of the waterfall plots. I mean, what are the response rates? And what are the durations of those responders?
Yeah, so the top line data shows us that the response rates for both TKIs are in the order of approaching 60 to 80%. For larotrectinib, the selective TRK inhibitor that was explored in 3 different trials across ages, as we mentioned, the objective response rate was 79%. And for entrectinib, that was also interestingly explored in the regulatory data set in 3 separate trials, the objective response rate was 57%. And beyond the waterfall plots, which I wish we could show you a picture, but you'll see that the majority of patients have disease regression. We have many patients with complete responses to therapy. And if you look at these waterfall plots, actually color-coded by the different cancer types, and they're very colorful because you see different color types go down on the waterfall plot, just highlighting that there are many different cancer types that responded to treatment. And another thing I'll call out is that beyond the impressive responses, the durability of these responses was also remarkable. And if you look at metrics like median progression-free survival for larotrectinib in an updated data cut that was a little over 35 months, that's amazing, almost 3 years.
And I think that's so important because every physician has a way of approaching an advanced solid tumor. And we rarely cure metastatic or advanced solid tumors. So it's important because colon cancer doctors look at their disease a certain way, and breast cancer doctors look at it a very different way. How do these TRK inhibitors fit into the current treatment algorithms for various solid tumors? Have you seen some confusion about how people are approaching this? Because it doesn't fit neatly into one disease only.
Oh, absolutely. And I would say that part of this also is that the labels, obviously jumping off the populations that were treated in the trials, so patients who had gotten many other therapies like chemotherapy, perhaps immunotherapy in the past. However, when I approach picking a treatment for my patients, it's all about the odds of benefit. And in the lung cancer world, I can say that compared to some of our other treatments, the response rates, the progression-free survival that you get with a TRK inhibitor really beat out the outcomes that we see with many other systemic therapeutics. And so if you look at the NCCN guidelines for lung cancer, it's actually recommended that you start with targeted therapy. Maybe, George, you can provide your insights into other cancers like sarcomas and, weighing the activity of these targeted therapies against other systemic therapies.
Well, I think that what you just said for lung cancer holds definitely for sarcomas, where we have therapies with modest activity at best in general. And if we find a driver like a TRK fusion, there's absolutely no question. If this were my family member, I would want them treated first with an NTRK inhibitor. And I can't tell you how many patients have gone through multiple lines of chemotherapy for various sarcomas without getting molecular testing. We get the molecular testing and find a TRK fusion, and then they finally get this drug. And it's just remarkable how fast it works and how well people feel on it. I mean, that's the other thing. If this were some really toxic drug with lots of bad side effects, it would be a very different approach about considering risks and benefits. But my experience is that these are extraordinarily well-tolerated drugs. And I think that's your experience as well. Is that right?
It absolutely is. And even if you look at the numbers, the rates of dose modification, dose reduction are really low compared to many other tyrosine kinase inhibitors. And in terms of overall side effects, we're really not seeing much. Most of them are grade 1 or 2, but really, if you treat anyone with this, they'll tell you that they feel much better on a TRK inhibitor in general than they have with, let's say, chemotherapy if they've gotten that in the past.
And Alex, you're lucky. You're a lung cancer doctor, which is something we never would have said 30 years ago. But now there's so many molecularly targeted targets that are very effective in non-small cell lung cancer that all you're doing is spinning the wheel of fortune with the genomic testing, and then something has a pretty good chance of popping up. So how do you address lung cancer patients? Because with these drugs, especially with entrectinib, you've got two shots on goal. You've got an NTRK fusion, but you could also have a ROS1 abnormality.
Yeah, I think that, well, one, it really underscores the need for paying attention to the test that you use and making sure that's comprehensive. And we mentioned earlier that multiplex testing is the way to go if you're looking for a variety of things. But talking about perhaps side effects with agents that are more selective, obviously you can see a cleaner side effect profile. I wouldn't say that entrectinib is in the ballpark of other MKIs or multikinase inhibitors like cabozantinib or vandetanib that really come with a lot of other baggage mediated by the inhibition of other kinases. However, I think that because there are certain differences in the properties of these drugs, such as entrectinib perhaps getting into the CNS a little bit better preclinically than larotrectinib. Some of the unique side effects that were mentioned or intimated actually in the first session that are mediated by the inhibition of TRK A, B, and C, like dizziness, ataxia, increased appetite, weight gain, paresthesias. I've heard some say that they might be a little bit more pronounced with entrectinib, but overall not really a major difference. It's not like if someone reached for one over the other, that would be a mistake.
Yeah. Now, we're lucky in sarcomas. Rarely do sarcomas go to the central nervous system, but in lung cancer and many other carcinomas, they do go to the CNS a lot, and especially primary brain tumors obviously start in the brain. So what about CNS penetration activity with these drugs?
Yeah, as I mentioned, there might be differences on paper in the proclivity of entrectinib to get into the CNS, but in practice, in patients, both drugs have achieved confirmed responses in the CNS. So with entrectinib, the objective response rate intracranially is 50%. With larotrectinib, it's 60%. Small numbers, the confidence intervals overlap. And we see durable responses intracranially as well. So I don't think that there's a preferred agent in patients who have CNS metastases. As we've seen, both drugs work very well.
And I think that just shows more confidence in how important these drugs are for these fusion targets. The targets are so sensitive to them, is that even though larotrectinib was not designed like entrectinib to actually get into the CNS, it seems to work. It's remarkable that we have two good drugs for these previously untreatable cancers.
Yeah, that's right. And my mentor, Mark Chris, always says, if it's a good drug, it'll work everywhere.
Well, thanks for the discussion, Alex. I think this summarizes nicely where we are with this first generation of TRK inhibitors, and we're going to be talking about next generation ones soon.
Thank you very much, Dr. Demetri and Dr. Drilon, and thanks to you, the listeners, for joining us. As a reminder, to view the full program, The Expanding Clinical Application of TRK Inhibitors in Cancer Care, a podcast series, and to download the slide set associated with this discussion from the Clinical Care Options website, please click on the link in the show notes. As always, thanks for listening.